بسم الله الرحمن الرحيم. السلام عليكم. تكلمنا المرة الماضية عن النيرفس تيشو وتكلمنا عن myelinated و non myelinated nerve fibers. حكينا إنه المايلينيشن عادة within البروتين stain H and E stain usually المyelin dissolved due to the lipid nature of the myelin. حكينا أن المyelin is composed of phospholipid, protein and cholesterol. During H and E stain or protein stain usually it dissolves. For that it appears as a white area, and in the middle we have this is the axon. For that we use another type of staining which is called osmotoxation, and this osmotoxation stains the lipid. As seen in this micrograph, we can see the lipid is stained in a dark brown area. So this dark brown area representing the myelin sheath. In the middle, the white area represents the axons. والمyelin is present all through the myelinated axons, and within the peripheral nerve, myelin presents an internodal segment. Myelin is absent from the node of Ranvier. Node of Ranvier, it contains lots of mitochondria. It is the site of the saltatory or the jumping of impulses. Also, it provides the collateral branching or collateral axons. تكلمنا كيف الميلين بيصير. حكينا الأكسون invagination within the Schwann cell. Schwann cell start to wrap the axon. Initially a mesaxon formed, and then it continues to wrap around the axon until the axon is completely myelinated. So this is the axon and this is the membrane of the Schwann cell, while this is the nucleus of the Schwann cell. And this forms an inner mesaxon, which is the initial mid of the membranes of the Schwann cell, and an outer mesaxon, which also represents the mid of the membrane of the Schwann cells. Longitudinal section, as we can see, this is the axon, and those layers representing a longitudinal section, the membrane of the Schwann cell or the myelin sheath. The myelin sheath usually absent from the internodal segment. Also, there are some locations which are called Schmidt of Lantermann clefts, as we can see those spaces, and usually those spaces forming cleft. This cleft forms an arrowhead. This is the arrowhead. Those are called the Schmidt of Lantermann clefts, where the membrane of the Schwann cell is not completely opposing each other. In this section, we can see those are the node of Ranvier, the opposing for interdigitation, and as we can see the node of Ranvier, we do not have a myelin sheath, as we can see, and usually it's rich in mitochondria and it lacks the myelin sheath. On the other hand, we discussed the non-myelinated fiber. Still within the non-myelinated fibers, we have a Schwann cell, and this Schwann cell can form clefts which can engulf the axons of the neurons, and usually those axons is not completely round or not many layers. The membrane of the Schwann cells is not wrapping the axons into many layers, so those nerves are called non-myelinated nerve fibers. Those clefts can contain a single axon or a multiple axons. Several axons could be enclosed within the Schwann cell. Again, those are non-myelinated fibers. حكينا example of those. It is the post ganglionic fibers of the autonomic nervous system. Again, myelinated and non-myelinated fiber. This is the myelinated fiber, and while this is the non-myelinated fiber. Still in both we have the Schwann cell. Electron microscope. ثم انتقلنا to the connective tissue which wraps the peripheral nerve or the nerve trunk, and we said that single axon is surrounded by a connective tissue which provides the blood supply for the axons, which is called the endoneurium, while a fascicle or a group of axons usually is surrounded by a connective tissue which is called the perineurium, while the whole nerve is surrounded by the epineurium. Under the microscope, this is a peripheral nerve or nerve trunk in the 4 sections. Those 2 sections, H and E stain, while those are osmium fixation and silver stain. As we can see, if you see this micrograph, it is a peripheral nerve or nerve trunk, and those rounded small areas representing the axon within this peripheral nerve. As we said, in H and E stain, in both sections, usually axon in the middle, and the white area which surrounds the axon representing the myelin sheet, which dissolved during the preparation. For that, we use this osmium fixation to stain the myelin sheath. So those are axons within a peripheral nerve, and those are multiple axons or several axons within this peripheral nerve. Connective tissue which surrounds a single axon is called endoneurium, while which surrounds a group of axons is called the perineurium, and the epineurium surrounding the whole nerve trunk. Again, at higher magnification, we can see this is the myelin. In the middle, we have the axon, epineurium, connective tissue which surrounds the whole nerve trunk. Endoneurium surrounds a single axon, while a group of axons is surrounded by a connective tissue which is called the perineurium. Then we speak about the axonal transport system. Again, this axonal transport system is facilitated by the neurotubules. The main function is to transport materials which are necessary for the nutrients and the materials or the substances which are secreted by the neurons, mostly the neurotransmitters. Also, the axonal transport system can get rid of the wastes of the neuron. And we said that it is anterograde, which means from this pericardium towards the periphery, or retrograde, from the periphery towards the pericardium. And the retrograde usually carries the waste from the periphery of the neuron towards the pericardium. And usually this is the retrograde axonal transport system, the way of transmitting, or the way by which viruses and microorganisms affecting the neuron. On the other hand, the axonal transport system is divided into slow axonal transport system with low velocity and fast axonal transport system. Slow axonal transport system only travels in a single direction, which is anterograde, while the fast axonal transport system, it is of high velocity and it goes anterograde and retrograde, which means in both directions. And usually this is the way of the transporting the neurotransmitters. ثم انتقلنا إلى supporting cells. حكينا those supporting cells, they form a major composition within the nervous tissue, and they do not provide any impulse transfer. They are not conductive tissue. They do not have any action potential. They are able to divide, unlike the neurons, for that they are the sites of tumors within the nervous tissue. They do not form synapses and they do not have myelin sheath. وبدأنا في supporting cells within the peripheral nervous system. حكينا the first one is the Schwann cell, neurolamellar cells, and those cells provide the myelin around the axons. Each one cell within the peripheral nervous system forms an internodal segment which is bounded by node of Ranvier from both sides. Myelin is absent from dendrites, axon hillock, node of Ranvier, and the terminal ends of synapses. Those are the locations which are lacking the myelin sheath. The second supporting cells of the peripheral nervous system is the satellite cells. Those cells, they provide electrical insulation for the cell body because they surround the neuronal cell body or the perikaryon, and they provide metabolic exchange. They do not provide myelin. حكينا in this diagram, this is the Schwann cell to provide the myelin sheath, while those small cells representing the satellite cells in cross sections, they are cuboidal cells, surrounds the perikaryon. As we can see, those are the satellite cells which provide electrical insulation for the perikaryon or the cell body. ثم انتقلنا إلى supporting cells of the central nervous system. There are 4 types of supporting cells within the central nervous system: الأستروسايت or الأستروجليا, oligodendrocytes, microglia, ependymal cells. Both the astrocytes and oligodendrocytes, they are large cells, for that they are called macroglial cells, while the microglia and ependymal cells, they are small cells, and those cells usually forms about 40 to 50% of the volume of the central nervous system. Astrocytes, they are star-shaped cells. They participate in the blood-brain barrier. They maintain extracellular fluid, and they can remove waste, and they can scar forming cells. They provide scar formation, and usually they are participated with the blood vessels through a foot-like processes. Those cells are of 2 types: protoplasmic astrocytes, usually this is the protoplasmic astrocytes, gray, present in the gray matter, very branching, very thick cellular processes, has a rounded cell body. On the other hand, fibrous astrocytes only present within the white matter, thin cellular processes, not so branching, with polymorphic cell body. Also, they participate with the blood vessel. Again, they are only present within the white matter of the central nervous system. Those are the astrocytes, fibrous astrocytes, and this is an astrocyte with immunostain. Second type is the oligodendrocytes or oligoglial cells. Although those cells has cellular processes, but they are not so much like the astrocytes. They provide the myelin sheath of the axon within the central nervous system. They are analogues of the Schwann cells. The only difference, beside their location, they differ from the Schwann cells that they can give myelination for more than one axon. As we can see, this is the oligodendrocytes provide cellular processes to more than one axon to provide the myelin sheet which surrounds those axons. This is an oligodendrocytes. The third type is the microglial cells. حكينا those microglial cells, they are the macrophages of the central nervous system. They contain cellular processes, and they are either originated from, some textbooks says that they originated from ectoderm, from the neural tube, which means they are originated like all the nervous tissue cells, and others says they are originated from the monocytes. Those are the microglial cells, and they are the smallest cell of all central nervous system cells. The final type of cells is the ependymal cells. Ependyma, they form the ependyma, lining epithelia tissue of the cavities within the central nervous system, such as the central canal and the ventricles within the brain. In general morphology, they are cuboidal to columnar with cilia. They are ciliated epithelial cells. They participate to produce the cerebrospinal fluid with capillaries, and those called the choroid plexus, and usually they are bathed with the cerebrospinal fluid, which is of their production. In this diagram, we said it represents the whole cell types within the central nervous system: the neuron, oligodendrocytes, protoplasmic astrocytes, fibrous astrocytes, the microglial cells. Those are the microglial cells, and those are the ependymal cells. Finally, we move to the organization. حكينا إنه spinal cord is segmented into 31 segments: 8 cervical, 12 thoracic, 5 lumbar, and 5 sacral, with 1 coccygeal. And all of these segments has a white matter and a gray matter. The white matter, no pericardium, while within the gray matter we can find the pericardium. From the dorsal root, they receive the afferent fibers from the sensory fibers from the dorsal root ganglia. Sensory fibers, cell bodies موجودة في الdorsal root ganglia, then they send their stimulation towards the dorsal root, while the ventral root or horn of the gray matter contains the motor neuronal cell bodies, which give order to the targeted tissue, for example muscle, to contract. Those are the efferent fibers. So any nerve contains both sensory and motor fibers. In this live section we can see gray matter, white matter, dorsal fibers from the dorsal root ganglia. This is the dorsal root ganglia, and this is the efferent fibers or the motor fibers. Both fibers are merged to produce the spinal or peripheral nerve. The peripheral nerve, as we said, contains both motor and sensory fibers. In this section, histological section, stain section, gray matter, white matter. Within the white matter, no cell bodies, while within the gray matter we have the pericardium or the soma of the neurons, and those are the dorsal root. Both sides are identical, right and left, while those are the ventral roots of this spinal cord segments. ثم انتقلنا إلى peripheral nerves وحكينا they are classified into type 1 fibers, type B fibers, and type C fibers. Type A, they are myelinated. They are thick fibers with large diameter and the conduction speed is very high, and those types of fibers present في skeletal muscle and sensory nerves, while the type B fibers also they are myelinated with less diameter. طبعا احنا لما نحكي more diameter it means more conduction of impulses, the velocity is higher. type B less diameter also they are myelinated. Those types are present within the preganglionic autonomic fibers, while the type C fibers, the final type, those are unmyelinated fibers with the least diameter and the least conduction speed. Those type of fibers are found within the autonomic and sensory fibers. ثم انتقلنا للجانجليا. حكينا النيرف جانجليا هي عبارة عن aggregation of neuronal cell bodies outside the central nervous system. ومر معنا مثال اللي هو dorsal root ganglia أو spinal ganglia, which is a sensory ganglia, and the second type is the autonomic ganglia. طبعا في فروقات بين السباينال والautonomic ganglia. dorsal root ganglia usually located near the spinal cord, while the autonomic ganglia most cases it is diffused within our body. لما نحكي autonomic it means sympathetic and the parasympathetic ganglia. The spinal root ganglia usually it has a thick connective tissue capsule which surround it, and the type of neurons are pseudo unipolar neuron or unipolar neurons. Those neurons usually located on the periphery of the ganglia. In the middle usually we do not have neuronal cell bodies, so they are not fairly distributed all through the ganglia. They are pseudo-unipolar, they have no synapses, and usually they are myelinated fibers. We can see lots of satellite cells surrounding the pericardium. On the other hand, the autonomic ganglia, usually this type, is diffused within the walls of the organs, for that it is called intramural, like the GI and the urinary bladder. Those ganglia usually they do not have capsule. Some occasions some ganglia contains a thin connective tissue capsule. There are differences between the dorsal ganglia and the autonomic ganglia. As we said, within the autonomic ganglia we have synapses. Usually those types of neurons form synapses. في dorsal root ganglia we do not have synapses. The type of neurons في autonomic ganglia multipolar neurons, while في dorsal root ganglia it is unipolar neuron. The density of satellite cells is less than the dorsal root ganglia, and usually axons are unmyelinated axons. The final difference is that the neuronal cell body are diffused all through the ganglia. They are not aggregated on a part of the ganglia. Those are generally the differences between the two ganglias. في الspinal في الbrain we only have to know that we have gray matter and white matter. Gray matter is from outside, unlike within the spinal cord, which is in the middle, and it forms the cortex within the brain, while the white matter is called the medulla. And the final topic is the injury of the neuron. حكينا الانجري of the neuron, either the damage is on the cell body, pericardium, or the axon. If the damage in the cell body, this will cause damage and death of the neuron, وبالتالي this will cause also atrophy of the innervated tissue of this neuron. Also, if this neuron forms synapses with other neuron, it forms a transneural degeneration, which is the degeneration of the synaptic neuron. This is if the damage on the cell body. The other situation if the damage was at the axon, let's say at this location. Initially, this damage will cause a degeneration which takes place for 2 to 3 weeks, and then a regeneration takes place after the third week. Within the degeneration process, we have anterograde degeneration, which is called the Valerian degeneration, and this takes 2 to 3 days, usually affecting the distal part of the neuron. This causes a complete degeneration of this part, which is called the anterograde degeneration. This part, anterograde degeneration or Valerian degeneration, and the debris will be phagocytosed by the Schwann cell, microglial cells, and the blood monocytes. Then we have another type of degeneration, which is the retrograde degeneration, which is affecting the proximal part of the neuron, this part, which involves 2 internodal segments. Meanwhile, during this, as we say, if the injury towards the axon, a cell body or the pericardium has its own changes. Cell has swelling, eccentric nucleus goes to the periphery, and the disappearance of the nasal bodies. As we recall زي ما بنتذكر انه حكينا بصير any damage النيورون بصير عندنا nasal body reaction او الكروماتوليسيس او the nasal bodies, disappearance of the nasal bodies. Those are the degenerations which are occur within the neuron. Then after that, a regeneration will take place. شو بصير في regeneration؟ As we can see, regeneration will start at the third week. Activity بصير regain neuronal cell body أو pericardium will regain its activity, appearance of nasal body, appearance of the normal size of the pericardium. Then the Schwann cell will start to form bridges, and those bridges will guide the healing process for the axon. Those axons will create a growthing parts أو growth بصير فيهم. Those growth are called sprouts, or they are called neurites. Many neurites are formed. Only few of them will manage to reach towards the targeted tissue. Once it regains its original shape, then the neuron is completely healed. الآن زي ما حكينا, some of those neurites will degenerate. If the injury site is so far, then the healing will interrupted, and this healing will cause the axon at the end, it will not reach its target or the targeted tissue. فبالتالي this will cause a neuroma within the location of the injury. فبالتالي شو بصير للtargeted tissue؟ بصير له atrophy. This is the summary of the last lecture.